Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. This is Dr. Habib, and I am here with JP. How are you?
Doing very well, thanks.
Amazing. Our topic today is on partial reprogramming for anti-aging, and we're going to get into a brand new kind of unique thing that's come to the forefront of longevity medicine, anti-aging medicine, and that is the Yamanaka factors and the work that's been done over the past couple decades, probably the last 10 years heavily, and how it's accelerating as quickly as it is with new companies and new ways of completing some of these really interesting tools. And we're going to let you kind of guide the conversation today, and I'm going to help to bring in some topics along the way as we go. But this is a topic that you're really interested in, and I think it's something that our audience needs to learn about because anti-aging medicine is the next trend in medicine as it stands. There's no question about it. It is happening as we speak in real time. And so this is something that we need to talk about and get into. I guess the beginning where it became apparent to all of us in the world here that something had been discovered by Dr. Yamanaka. Let's start there.
Yeah, Dr. Yamanaka, Shinya Yamanaka, is a Japanese scientist. He's actually a physician. And the work that he did back in 2006 and 2007 had, frankly, effects beyond just science. It affected politics, believe it or not. For those of you who are old enough, you probably remember back in the 90s and early 2000s, there was a lot of debate in the public arena around the use of embryonic stem cells. These stem cells that were, pluripotent, omnipotent, that could differentiate into all sorts of different types of tissues. And the thought was that they would be sort of the next wave of treating really difficult to treat diseases. People were going in for stem cell treatments and had to go to different countries. And it was very, very controversial because the source of those was typically aborted fetal tissues. And Shinya Yamanaka stepped into the fray, if you will, and began looking to see, well, is there a way that we could reverse the process that naturally occurs where cells go from a stem cell to a more differentiated progenitor cell and then became the somatic, fully differentiated cell, like a skin cell or a liver cell or a endothelial cell, et cetera. And what he did was identify, in what was an incredible amount of work, identified that there were 4 proteins that in these pluripotent stem cells exist, but then become epigenetically silenced as you get closer and closer to a fully differentiated cell. And that if you could unlock those, you would actually have the cell go backwards in its differentiation state all the way back up to being that stem cell. And so he created an entire field around what are referred to as induced pluripotent stem cells. And these are cells that are, through literally triggering these proteins to be expressed, causing these stem cells to come into existence from a skin cell or from a blood cell. You take that cell and you activate these Yamanaka factors, and there's 4 proteins. They are OCT4, OCT4, SOX2, SOX2, CLIF or KLF4, and the protein called c-MYC, which is C-MYC. And these 4 proteins, when active, are involved in the differentiation process and proliferation of cells, et cetera. So really, really fascinating that we're already programmed with them. And he showed that by activating them, you could reverse the age, literally the age of the cell, and then give it more stem cell-like activity.
I love this because it's almost natural thought process when there is a political issue, when there is some controversy about bringing in embryonic stem cells from a different source, from somebody else, right? A different genetic and biologic breakdown, we're now looking at what are the factors that those cells have that our fully differentiated cells don't have. And the work that he did essentially allowed us to identify these 4 factors that, when turned back on, as you mentioned, are going to allow your own cells to reverse in age, to actually go backwards in age potentially. And so that's where this is coming down to. So the idea of then adding in embryonic stem cells goes away, because we're not worried about other people's genes, and we're not worried about any potential political or ethical/moral issues that come up with using somebody else's cells. So that's really great. Let's talk about these 4 factors, the OSKM or the Yamanaka factors. What kind of work went into identifying them? You mentioned that there was a lot. I don't want to go into obviously the depth of that, but I want to understand what these 4 factors do or what their intention is.
Yeah, and you're right. Let's not dive into the details of what he did, because it was truly a tour de force of scientific effort, especially given the technology that was available at the time. I think somebody walked up to Richard Feynman once at a party and said, can you describe what your Nobel Prize winning work was, in a sentence or two? His response was, if I could do it in a sentence or two, it probably wouldn't have been worth a Nobel Prize. And same thing is true here, because in fact, Shinya Yamanaka did win the Nobel Prize for the work that he did. So again, these 4 proteins are involved in a lot of different activities within the stem cells. They're involved in proliferation. They're involved in identifying where what should be epigenetically silenced in a stepwise function. They're involved in a whole host of early differentiation steps.
Okay.
But I think it's one of the things you said. I just want to make certain that our audience understands. Just because you take a cell, a skin cell, and you reverse its differentiation, that doesn't necessarily mean you're making it younger. Okay. And that's really the trick here, because the next step in the process was to, people said, well, okay, great. We can use these OSKM factors to make animals younger. Well, that's not exactly what happens when you do this. When you activate all 4 of these proteins in a fully developed animal, an aged animal, what will happen is you'll see some anti-aging effects, early on. But as those cells de-differentiate, well, they're not doing the things that skin cells need to do. They're not doing things that liver cells need to do anymore, because they're not liver cells anymore. They're not skin cells anymore. And what you end up with is really non-functioning tissue that has lots of potential to turn into other things, but it hasn't done it. And so the animal doesn't survive. And to the extent that you stop it along the way, that process along the way, because you can stop it, if you do, you might have some of those cells so de-differentiated that they'll begin to differentiate into other things, and you get sort of unregulated growth. And that unregulated growth, that's cancer. So it's really interesting in terms of de-differentiating individual cells, but entire organisms, no, that's not going to work. You're not really going to make them younger. You're going to have really a lethal effect on them.
Mm-hmm.
But that's where a second group of researchers stepped in. One of the leaders in it was a Spanish guy by the name of Juan Carlos Espesua Belmonte. And what he recognized was that this process of de-differentiation did have 2 steps to it. It wasn't simply de-differentiating. It did have a rejuvenating effect initially. And that was responsible for that short-term sort of looking like there was an anti-aging effect before it moved into the de-differentiation phase. And so he identified, along with others, that if you only use the O, S, and K, or if you used all 4 of them, but only for a short period of time, just a few days, that what would happen is the cells, the first step in the process of dedifferentiation was actually rejuvenating them back to an earlier, fully differentiated, but an earlier state in terms of age. And that was really exciting. That was around 2016, 2017 that they did that work. And that's where you start to see animal studies where they've taken animals who were, the equivalent of 70 years old, and showed that instead of them living for just another equivalent of like 10 years, they live for another 30 years, because their fur goes from gray to more full and dark. Their organs actually start to become more rejuvenated. Their learning, their ability to think is actually improved, so that they're able to solve, little mouse problems that they challenge them with more effectively, more quickly. So really exciting, and the process is, it really hits on all of the really kind of key hallmarks of aging that we see. We've talked about those hallmarks of aging on an earlier podcast, but things like telomere shortening. Well, when you trigger these OS and K, you see telomere lengthening. So you're activating telomerase. When you see the epigenetic drift that occurs in aging, where the epigenetic marks either get degraded, or new epigenetic marks get put in place on both the DNA and on histones, those things sort of reset back to where they were. And you think to yourself, how does the cell remember where all of those methylation marks should be? Literally hundreds of thousands of them, if not millions of them, across the entire 3 billion base pair DNA. It's literally going back and putting them back where they were supposed to be, removing ones that shouldn't be there and replacing ones that have been lost. That process takes place in just a matter of days. And that means that you're reversing the age of the DNA. You're also affecting your mitochondria. It's activating autophagy. It's activating mitophagy. You're getting rid of things that are run down in the cells. It's really a remarkable cheat code, if you will, for rejuvenating cells.
That sounds really interesting. I'm going to just throw out an analogy here. I want you to imagine a really big, really thick textbook, and that textbook has Post-it notes and reminder slips posted on a million of these pages. You have a 3 billion page book, and you have a million Post-it notes listed on that textbook. And over time, you've moved these million Post-it notes. A couple of them have come off, a few 100,000 have been put in different pages, and it's a dynamic process. And that's the aging process by which you're understanding and you're utilizing this book in its way that it's been used. And what these OSK and sometimes M factors are doing is they're almost putting you in reverse, where they're saying, oh, you used to have a Post-it note on that page. Now you have it on this page. Let's take it back off and let's go and put a new Post-it note on the page that you used to have it on. And you remember which page that is. You're going back to page 21,142 and you're putting on a Post-it note because it used to be there, but you had taken it off. And so when your cell was younger, when it was in its child age, kind of your 10-year-old kid type of age, it had the Post-it note there, but when you're 60, it doesn't have it there. So what it's doing is it's saying, let's take the Post-it note off from this time when you're 60 and let's go put it on at the 10. But it does it in such a dynamic fashion and such an in-depth kind of process. And I'm hyper simplifying this, obviously. But the idea is you're going backwards to a time or an age pattern of epigenetic marks or Post-it notes in that book that used to happen when you were 10 years old rather than 60 years old. And that's happening in every cell of the body in an ideal timeline.
That's a good analogy. I use a similar analogy. I think about it as, you're working on a 747, okay, which has a million parts. And you have an instruction manual for putting that entire 747 together. And when you're a pluripotent stem cell, you want to have access to every page in that book because you're going to need to put together the whole airplane. Okay. That's like the guys who design it. They need to be able to look at the entirety of that book. But the people who are building the landing gear, okay, they need one chapter of that book. Okay. So what happens is during the differentiation process, there are a series of marks. They're methyl groups that are attached to certain base pairs of DNA, and stretches of DNA get lots of them. They're called CPG islands. And they have the effect of typically locking away that DNA so that it doesn't open up. Okay. And that way, it's sort of like gluing the pages of the instruction manual together. Okay. That section of the book, you don't need to look at. You just need to look at the pages. When you become a skin cell, you only need this DNA to look at. And so by the time you get to a fully differentiated cell, something like 75% of all of the genes are locked away. You're not looking at them. Okay. And 25% are available. Now, all cells need certain genes. So there's a set that are always open. But then you got the difference between a liver cell and a skin cell is that there's probably 5 to 10% of the genes that are exposed and available for liver cells that just aren't for skin cells. The problem is that as you age, you begin to lose some of those methylation ticks or marks, and some other ones get put on. So think about it. You're working on the landing gear. You've got, you're fresh out of school. You got your new instruction manual. It's just one chapter. That's the only chapter you're actually able to open because everything else is glued shut. But by the time you're 30 or 40 years on the job, well, you've spilled coffee on some of those pages. You've glued some of them shut accidentally with grease or whatever. And some of the other ones that were glued shut have sprung open. And so now when you open your book, it's not so easy to find what it is that you're supposed to be making. And that's really what happens. You get to the point where some of the proteins that are supposed to be made aren't being made, and some of the proteins that shouldn't be made are being made. And you get to this senescent state where the cell starts to realize, I'm not really functioning the way I'm supposed to. And it settles into this sort of, I'm not going to divide anymore. I'm not going to make any more of me. And I'm going to start to be a little bit inflammatory, and I'm not really doing my job right, but I'm sort of floating here like a zombie. And that's one of the things that when you replace all of these epigenetic marks in the way they're supposed to be, and you close off the DNA you're not supposed to be producing, and you open the stuff that you are supposed to be producing, that process rejuvenates the cell. It takes away. You no longer need to be senescent. There's less inflammation. There's less, and all of those things that go along with aging, inflammaging, all of that stops happening because you've restored what genes you're supposed to be making.
That restoration piece, I think, is so powerful to understand. And what a unique kind of finding here that these 4 factors, this OSKM, not even necessarily the M, just the OSK factors are particularly the 4 factors that have been identified to help with that repatterning to a pattern that is identified as being younger, or kind of cleaning up that book or cleaning up that manual. I think that's so cool. I had the pleasure of watching a presentation at the A4M conference. I believe it was last year, it may have been 2 years ago, with David Sinclair speaking. And he was talking about the use of these OSK factors that they were using. And they, in their lab, had repaired and reversed age, quote unquote, on an optic nerve. And I think this was really, really cool. I would love for you to dig into what progressed following Belmonte's work and the idea of application of these OSK factors a little bit more, and how researchers like David Sinclair are utilizing these OSK factors.
Yeah, David Sinclair is a really interesting individual, MIT grad up at Harvard now. working. His line of research is trying to make this more applicable to a wider audience and make it easier to do. Because when you're manipulating genes that are endogenously there, we're not snipping in new gene, or we're inserting new genes with CRISPR, et cetera. What we're doing here is activating genes in individual cells. And and that's what Shinya Yamanaka did and Belmonte did and others in individual cells. When you move to an organism and you start to try to do this, your efficiency level is exceptionally low because you have to use, a variety of different rather elegant and sophisticated tools to try to unlock those genes, because the cells have locked those genes down. We just talked about the epigenetic locks and silencing programs. So by unlocking those genes, you can do that in maybe 0.1% of the cells. And yet you still get the benefit. You still see the pretty remarkable benefits, but it's a very, very low efficiency process, and it's very difficult. And you can maybe do it on one tissue type. You can't do it across the entire organism the way you really want to. There's really two avenues, and we'll talk about both of them. One avenue, which David Sinclair is moving down, is saying, well, yeah, okay, these proteins, when we activate them, they go on with a cascade of other things that they trigger in the cell to do. Can we do that? Can we trigger that cascade, not with proteins that we have to activate in this using these sophisticated tools, but can we do them with small molecule drugs? Are there drugs that exist today that we can use to do this? And he's identified, along with other researchers. I mean, this research is happening in many different research institutions and companies, but he's at the forefront of it. And he and others have identified some things like forskolin and valproic acid. I mean, things that are used in medicine as medications. Valproic acid is used in for example, in epilepsy. These medications seem to be able to have the downstream effects that triggering OS and K have, but you can do it with small molecule drugs you can take with a pill or an injection. Now, there's others that are in the process of being created as new drugs that will have these effects as well. And they're trying to maximize their effect and minimize the off-target effects that they have. And you have that with small molecule drugs. I mean, you're going to have side effects. You're going to have off-target effects. And so the applications for those may be more along the lines of, yes, as you said, treat blindness, regrow the optic nerve or reactivate an optic nerve that's been damaged. That's really exciting work. Work in glaucoma, work in atherosclerosis, work in peripheral vascular disease, work in skin lesions, and just from a beauty standpoint, making yourself, less wrinkly, as you grow. These are all potential applications of this anti-aging technology with small molecules. Now, that's interesting, and I'm very encouraged by what I see there. But there's some new work that's been done, which I'm frankly been blown away by. I think it's absolutely amazing. And this is, part of the AI revolution. What the people at OpenAI did, and they did this in part because Sam Altman, who is the main guy at OpenAI, actually invested in a company called Retro Biosciences. And so Retro Biosciences has as their mission to optimize the OS and K proteins.
I just want to shout out that Retro Biosciences is such a cool name for a company that's involved in anti-aging. Like, what a cool name.
Yeah, there's no question that came right out of Silicon Valley.
Yeah.
Just cool stuff out there. So what OpenAI did was they took one of their LLMs, their top LLM, the 4.0, ChatGPT 4.0, And they took that as the base model and they began to feed it additional information, literally millions of documents and tokenized images of proteins, protein gene sequences, proteins, folded proteins, not just the amino acid sequences, but how they fold, sort of in the alpha fold model. And what they did was they gave it all of this information, and then they said to it, okay, O, S, and K. Here are the genes. Here are the proteins. Here's where they're active. Make them better. Give us better genes. Optimize them. Look at the direction that evolution is taking it and make it more effective. How can we increase from 0.1% of the cells expressing it properly? How can we make it better? And sure enough, ChatGPT said, okay. 4 seconds later, spit out, no, not 4 seconds later, obviously, but it spit out, hundreds of potentially optimized genes. And so then Retro Biosciences took this information, sequenced all sorts of genes and all sorts of proteins from this, and they found that, for example, with what they call retro socks, 30 to 40% of the proteins that were spit out by this large language model that had programmed were actually more effective than Mother Nature's SOX 2. And so when and I think they only did it with two of the three of the O, S, and K. I think it was they did it with S and K, and they kept Oct4 as the same as it was. And maybe they're still working on that. Maybe that's their secret sauce. I don't know. But what they have published just within the last couple months is that when they used the optimized versions of these proteins, that they can go from 0.1% of the cells expressing this to 30%. And that and it's a little bit confusing in what they published, but in some cases it's up to 85% of the cells are expressing this rejuvenated state. Now, it's really amazing in two fronts. Obviously, what an incredible increase in efficiency. I mean, they're advertising it as a 50x improvement, but when I look at 0.1 to 85%, it seems like an
higher
50%.
Yeah.
But the other piece of it that's fascinating is all they have to do is provide the proteins.
Yeah.
They don't have to insert the genes and then activate the genes with promoter sequences that they are triggering and controlling. So there's a lot of what appears to be a lot of technical difficulty associated with the original way of doing it that they've also circumvented. So it's exciting. This technology is moving in multiple different fronts into human clinical studies that are supposed to be starting this time next year. They're going to be enrolling patients.
It's pretty astounding to think just how the slope of the curve on this has accelerated as quickly as it has, right?
Exponentially.
Yeah, exponentially, literally.
This is the difference. Human beings don't naturally think exponentially. We're talked
very
about
linear, yeah.
We're linear in we think, if you have a 10-year time frame to do a project and you're not 90% done by the end of 9 years, then you're behind schedule. When that is absolutely not true. In fact, if you're even 20% done at 9 years, you're ahead of schedule, because the chances are you have optimized your process to the point where making the last 80% in the last year is you could do that on just 2 days a week. I mean, this is the trick of exponential growth, and that's why they talk about it in the AI world. They talk about this singularity event, where advances will start to become so rapid that everything happens now. That really there's no time delay between what you can think of and what's going to be possible, and then occurring and then deployed. And the time frame for this is happening so rapidly that as Ray Kurzweil said, and I like to say in some of my talks I talk about Ray and his he had a saying which was live long enough to live forever. The trick is stay alive, because if you make it to the singularity, you're going to be around for a long time. I mean, it's going to be kind of, I mean, I'm not saying we're 5 years away or even 2 years, 2 years or 5 years, but there's going to come a point probably in our lifetimes and certainly in our children's lifetimes. And I'm sort of hoping it's ours, where we can go in at age 70 and get a treatment that'll last for 4 or 5 days. And, you're not going to walk out looking like a 20-year-old after 4 days.
Well, some might.
But, well, no, well, that's a lot of plastic surgery. But no, now we're talking about like genetic surgery, where you're going in and you have this treatment, and you'll feel a little better right away. You'll feel really good within a week. By a month out, people are going, "Did you dyed your hair? You put on some weight? You look stronger? Been at the gym?" And then by 6 months out, people are thinking to themselves, "This is a 35-year-old guy, not a 70-year-old guy." And before you know it, you're back to 20. Because all of your cells have gone through this process and have rejuvenated. And then, and there's nothing else in your life that you won't forget everything. It's not like you're going to lose your memories. You're just going to lose the plaques in your arteries. You're going to lose the tangles in your brain. You're going to lose the fatigue that you feel when you go run. You're going to lose the aching in your joints. That is fascinating.
It really is. I want to ask the question, though, does that mean that at some point in the future we can treat our bodies like shit and then just go in for an OSK treatment and rejuvenate? In theory,
no, you're not going to, if you weigh 400 pounds, you're not going to come out weighing 180. You're still going to have to lose the weight, but you're going to be more capable of losing the weight. That's sure, for sure. I don't think that you're going to be able to cleanse your body of all the toxins. So you're still going to need detoxing programs. It's not an excuse not to exercise. It's not going to suddenly make your lungs. There's plenty of 20-year-olds out there who vape and need a lung transplant, or drink too much and need a liver transplant, or take too much Tylenol and need a liver transplant. Those things are still things you need to be watchful of. But it's going to give you a chance. I mean, right now we're really fighting, we're fighting the good fight, but it, until somebody doesn't, everybody dies. And so we're hoping that maybe the first person to make it to 1,000 years old is alive today.
I think that would be really cool. And I think just this point of living long enough to be able to see this happen, Ray Kurzweil's kind of theme here is important. I want to make sure that people are aware of the fact that this is happening, and yet it is not the cure-all for everything that we're already doing to ourselves, right? That as a society, or as individuals, forget the as a society piece right now, we need to still stand up and take control of our own health. We still need to put in the effort and put in the work that's required to build up our muscles and remove the toxins from our bodies, and try to live with as long and as healthy a health span as possible, right? That's a huge piece to the puzzle here. I'm excited to hear what you see over the next few years with regards to this particular therapy, and where do you feel like it's going to go? Are people going to be able to go in and, instead of needing like a liver transplant, they're able to have an OSK treatment on a fatty liver or a NASH type issue, and their liver is given a second chance to function well, or something along those lines?
Yeah, no, I think that is going to happen. And just to give a little context here, I mentioned earlier that while David Sinclair is certainly at the at the tip of the spear, at least out there publicly, with respect to a lot of research that's going on in the making of small molecules, the entire field is gathering a lot of interest and investment from some pretty smart people and some pretty wealthy people. So just to give you an example, um, Google. Google has a division called Calico, which is the California Life Company. Calico is all about getting involved in the anti-aging space. They're using their protein folding AI, which is this, very similar to this work that was done by Retro Biosciences and OpenAI. They have the ability to do very similar work, and I'm sure that they are. And they've got Google's finances behind them, and that's a lot of money. But there's also, some startups that are like Retro Biosciences where they're getting these tech billionaires to invest. And there's the company Altos Labs, which is where Juan Carlos Belmonte works, or at least is one of the lead scientists. He and his team got a tremendous amount of money from Jeff Bezos and other billionaires out in the Silicon Valley area because they look at this, and these guys are smart. I mean, yes, they have lots and lots and lots of money, but they also don't waste it. They believe that this is something that's worth their investment and their time and their support. And that company is called Altos Labs. So if you start looking at, and there's others. I think there's another company that I think David Sinclair's involved in. I think it's called Life Biosciences. So you put Retro Biosciences, Life Biosciences, Altos, and Calico, and others, and you realize this is at the forefront of pharmaceutical and biological sciences research that is getting a lot of funding and a lot of attention, not just from the NIH and National Science Foundation, because there actually isn't a lot coming from there. It's coming from private sources. Now you could say that that's ego-driven, or they want it for themselves, but I think they recognize there's a business play here that's real,
Yeah.
and I'm very excited to see it. I think the first things you're going to see are the things you were talking about. Things like, okay, I've got a fibrotic liver. I've got, potentially the need for a liver transplant. There's, 10 times the number of people on the liver transplant list than will actually get one. And gosh, that's a very, very, very invasive procedure and process. Is there a way to, with drugs, treat it so that I rejuvenate the liver that I damaged?
Yeah.
I think that's one. Optic nerve damage. We've already seen, animal studies in that.
The one that I see potentially as a huge market is pancreatic repair and rejuvenation with the diabetes levels that are out there, and
Yeah,
like that just
islet cell
being
transplant.
islet cell transplant type of
Yeah,
idea, right?
the question is whether or not, certainly with type 2 diabetes, where you've just fatigued those cells to no end. That I think is there's an opportunity to rejuvenate. Type 1 diabetes, I also think there's an opportunity, but I think you're going to need something to block the
Yes.
autoimmune. And actually, it's a little off topic, but there's a fascinating case that I just became aware of a woman who has a twin brother. And
You sent me this, I
if you,
remember.
well,
Yes.
if you look at her genetic, her genetic code, she has typically XX chromosomes in all of her tissue except her blood cells. Now, this happens sometimes in cases where people have had a bone marrow transplant. If you're a female and you get a bone marrow transplant from a male, well, your blood cells that are now made by that bone marrow are going to have XY chromosomes because those cells are being produced by the marrow that is from the donor who is male. But this is a situation where this woman, she's in her 20s, I believe, has her twin brother's genetic blood. So her bone marrow is producing is XY. Her liver, her, all skin cells, et cetera, all the rest of her body has XX chromosomes except her blood. And it has not caused her any problems.
Yeah.
It was found actually accidentally.
Yes.
Okay. And what they're recognizing is there an opportunity here to leverage this for tolerance building? How can we figure out exactly how that works gestationally so that we can then take that tolerance inducement where her body is not reacting negatively to the fact that she's got somebody else's blood in her. She's got somebody else's white blood cells. And is there a way to now induce that tolerance for organ transplants
That's awesome.
or at least bone marrow transplants?
For the sake of the listeners who don't understand this story, how it kind of came to, it has been posited, and this is not, they're not 100% certain, but this is the position that's been taken, is that during implantation, their placentas crossed of these two twins, and the placental.
Otherwise fraternal twins.
Yes, exactly. So these were fraternal twins, obviously boy and girl being separate genetic predisposing egg and sperm coming together, and they had implanted onto the uterine lining, but in some capacity, the thought is that the placentas crossed, and so there was a crossing or a creation of a bridge between the twins in utero, and what essentially happened is the boy's blood ended up going to her.
It was probably a progenitor of a macrophage. It was probably a macrophage progenitor that then went to the liver and then went to the bone marrow, but it was actually her brother's fraternal twins. It was her brother's macrophage progenitor that was transferred over and replaced in his, because they now share exactly the same white blood cells. It would be very interesting. It was not discussed in where we found this, but it would be very interesting to see whether or not, if you took a skin graft from the boy or the male and put it on the skin of the girl, whether or not it would be automatically tolerated.
That's a really interesting question, right?
Because clearly she is tolerating his blood and his white blood cells.
Yeah.
Is that tolerance now universal?
Transferred to all the other tissues, yeah.
I mean, twins, identical twins can do that because they have the same major histocompatibility complexes, et cetera. But this is a situation with fraternal twins where you wouldn't have that. But because there was this cross pollination, if you will, early on,
for lack of a better word, yeah.
but now he's not going to be tolerant of her, but she may be tolerant of him. So
we obviously didn't see this in the report, but did it say whether or not he had his own blood or her blood?
Oh no, he has his own. He has his own. Yeah. So
that's really cool.
They didn't swap. She just got his.
That's really cool. But being able to understand how that happened, how that worked, you're exactly right. How do we ensure that, or potentially utilize this information in future care ideas when it comes to longevity therapies and new organs being implanted into other people? So that's a really unique thought process.
But that doesn't mean that there's an infinite number of those organs around.
No.
And even if there are, take care of the ones you've got. Don't drink too much. Don't smoke. Don't do things that are risky behavior. I mean, put it this way: you're not going to grow a new arm. Okay, if you lose your arm in some stupid accident, you're not going to get that to grow back.
Yeah, we're not going full Wolverine and Deadpool rejuvenation, like rebuilding your tissues. We're not superheroes, and that's okay. But don't expect that to be a thing.
There are also a lot of societal questions that need to be asked and answered. What happens if you're born, you get into your 20s, early 30s, you get married, you have a family, and now you're at age 70 and your kids are at age 35. and you go in for this treatment, and now you look like you're 20 and your kids look like they're 35, and then they become, at what point do, you say you marry for life, but that the question, reasonable question is, you know, there's an expectation when you get married that that's like 50, maybe you'll make it to 50, 60 years. That would be fantastic. Do you want that for 600 years? Do you want that
Yeah,
for 1,000 years? You know, I, I'm not saying I wouldn't, but I'm just saying that it's going to be an interesting game. And the question is, do you want to have another set of kids? Do you had
very
one set of
interesting.
kids? Do you want to have another set of kids? It's and what if one decides, no, I'm not going to do that. You know, it's
Instead
going to
of
make
till death
it.
do us
There's
part, it's going to be till OSK factors do us part.
there's a lot of gold diggers out there, and then male or female gold diggers who are 25 years old and they're married to a 75-year-old, thinking, "Oh, I only have to make it another 10 years, and then I'm going to inherit." What happens now? He's 25 again, also.
Yeah.
You didn't really like him that much. It's going to be a problem.
There's going to be a lot of fun societal
You can tell I've been
issues that
thinking about
pop
this a
up.
little too much.
This has been a lot of fun. Really interesting stuff here. The OSK factors, Yamanaka factors, are very clearly the consensus way that longevity and anti-aging are going to be implemented over the next decade or so, it sounds like. But who knows, with AI and the ability for us to process information as quickly and as effectively and efficiently as we are now, we're in an exponential push, and we've always been, but we're on a heavy upswing right now on this amount of information and processing capabilities that we have. I want to drop you one last opportunity to just kind of throw out what do you feel like is the future of this and the application of this going forward?
So I'm going to say two things. One is, I think the initial things you're going to see are probably on skin and the optic nerve, because they've already gone there, and eye treatments for glaucoma and other things. I think your ideas for things in the big three of cardiovascular disease, metabolic disease, and I think lung disease, things like emphysema, et cetera. I think you're going to see those be treated. Probably musculoskeletal issues as
Mm-hmm.
well, like osteoarthritis. But I think that all of those things are going to converge on the overall organism rejuvenation therapies that I don't think are more than a decade away from at least at least uh, large scale clinical studies, if not approvals. But in the interim, we had another podcast episode where we talked about what is the autonomic nervous system or autonomic deregulation theory of aging,
Yes.
where what we see in all these hallmarks is that they get worse the more stressed you are, the more you're living in a chronic state of sympathetic activity and failing to get back to that parasympathetic state regularly. This isn't going to make you less stressed. It's not
That's
going to make you
exactly
more
right.
resilient. It's not going to, just because you get more life and more youth doesn't mean that you're going to spend it wisely and enjoy it. So what I would suggest is that you adopt those really good habits right now to get back into parasympathetic recovery regularly, so that you can not need this therapy as regularly, um, and that you do what Ray Kurzweil said, which is live long enough to live forever.
I love it. My final kind of thought here is I see this as the inflection point on medicine. I think medicine and pharmaceuticals, as it has been practiced, and uh, kind of the way that we've been living in this pharmaceutical revolution for the last few decades now, has very much been in symptom management, symptom suppression. And I think we're at the crux or the inflection point of a massive shift towards this anti-aging, longevity-based medicine, where it's meant to actually repair and heal. It's not necessarily curative to disease, but rather providing your cells an opportunity to recover and heal, and give you a second chance and potentially a third or fourth chance at doing certain things. And I feel like that's where the shift is coming. So that's at least what I see happening over the next little while. And as new updates and new research comes out, we will definitely be sitting down and sharing our new findings and the new data points with all of you, because that's what we want to do here at the Health Upgrade Podcast. What can we do to help you upgrade your health? This has been a fun episode, I think. JP, any final thoughts?
I do have one, which you just touched on a little bit, and I want to just run with for a second, which is that a lot of what's being done right now in anti-aging medicine is not considered by the regulatory authorities to be medicine from the standpoint of something that they're going to regulate. Okay. And I mean that because the FDA does not consider aging to be a disease. Okay. And there's pluses and minuses. Your first instinct is, if you're like me, your first instinct is great. Don't have them regulated. Let's figure this out. Let the market decide. But by the same token, regulation has its place.
It does.
And I think we need to find, and maybe it's a new category. I mean, listen, the FDA started out just looking at drugs and food, but then they got biologics, and then they opened up a branch for devices. Maybe there needs to not try to jam this what I sort of consider this, health sustaining, and youth sustaining.
Mm-hmm.
Maybe we need a separate division within the FDA that regulates these preventative or health sustaining. We're not treating disease, but what we're doing is we're preventing disease. We're preventing the breakdown. We're preventing degeneration.
Or reversing, yeah.
And maybe we call it the rejuvenation center. They got the radiological and devices center. We got a rejuvenation center. I don't know.
Interesting.
But I think there's going to be some sort of governmental regulation over this process to make certain that it is safe and make certain that the research is being done well. Because something this exciting, something as exciting as, because we're really talking about something that's pretty close to the fountain of youth.
Yeah.
Something this exciting is just an opportunity for people to abuse.
Yeah, the bad actors are for sure going to be out in full force when this happens. So that's where regulation can come in and be an actual positive tool for society to use to protect us from those bad actors. So I see that the value there is for sure.
Yeah, so that's a little outside the science world. I'm putting my lawyer hat on now. So just if
you're allowed to, you did complete your JD, so that's a big deal.
No, but I guess my point is, for the listeners out there who are hearing this, think about this. Think about what your own opinion is. Think about it. You gather some additional knowledge, do some research on your own, and let your opinion be known to your congressman or other people that are in the government, that if you have access to it, and tell them, like, hey, I think this needs no regulation, or I think this needs this type of regulation. I don't think it should be blocked. I think people should have access to it. And there's a lot of other issues that have to be thought through, and moral, ethical, societal, economic, all of these issues need to be addressed. But the good news is that healthcare is a really, really big expense in the United States and around the world. And a lot of it has to do with care for age-related diseases. And if these age-related diseases can be avoided through this process, think of the economic boom that will be delivered to society. So it's not just, oh, I get to live, for 1,000 years. It's, hey, maybe we're not going to go bankrupt. Maybe we just saved $3 trillion a year.
Yeah.
In a couple of years, that adds up to some real money.
It really does. I love it. This was a really fun topic, really great discussion here. So thank you for bringing this one up for us. And I think this is the future of where we're going in the majority of healthcare space. So keep your eyes on everything Yamanaka factors, OSK factors, and anti-aging, because something is changing here for sure. I hope this has been a great episode for you guys to listen to. Thank you for getting to the end here. And if you enjoyed this one, share this with one person who you think could utilize this information to upgrade their health. We'll catch you all on the next one.